Transcriptome refers to the complete RNA repertoire of the cell where we can have different kinds of RNA, mainly it's messenger RNA. So that messenger RNA is actually where we can study the gene expression, and we can study the function of the genes through this messenger RNA. Same way we can have other RNAs which are non-coding RNA like transfer RNA, ribosomal RNA, we can also have small RNAs, and we can also have microRNAs. So all of them, they are studied in, we study them in transcriptomics. So transcriptome sequencing can be what we call it as RNA sequencing, where we study the messenger RNA mainly. It can be microRNA sequencing, deep SAGE, deep CAGE. These are the old techniques, but now they are replaced by the SAGE has been replaced by RNA sequencing or RNA-seq. So that was also focusing on capturing the messenger RNA or the expression of the genes in terms of messenger RNA. We can also study the polyadenylation sites, the sites where poly A tails are attached to messenger RNA. That is also included in this transcriptome sequencing. So here in this slide, we see this transcriptome sequencing in detail. We start from here towards 5' end. So if we are capturing the 5' ends, there is a technique called as CAGE, that is the cap analysis of gene expression. So in this way we can see what we have towards this 5' end of this messenger RNA. Normally it is used to study the modifications around the promoter and transcription initiation site regions. RPF is obviously this RNA that is coming from the exons, then does protein coding. In protein synthesis, it interacts with the ribosomes. So if we are studying those ribosomes, profiling methods, RPF, so in this way we can study the RNA which is involved with the ribosome if we focus on that region. Whereas RNA-seq is where we study the whole messenger RNA. SAGE is serial analysis of gene expression. So that is mainly focused towards the identification of these RNA transcripts towards 3' ends using some specific tags. Whereas polyadenylation sites, they can be study what nucleotides are there where we have this poly A tails are attached to messenger RNA. So PAS focuses on studying that part of the messenger RNA. So in transcriptome sequencing, we see that there are different steps. So first of all, we prepare our samples. That process is called as sample preparation. We submit those samples for next-gen sequencing, and then after we get our sequences, what we can do with them is we can do a lot of data analysis where we can map those short fragments, we call them reads, to some reference if it is already existing there, or we can put them together, assemble them together, and we can have the even transcriptome assemblies. We can, once they are aligned to those references, we can do visualizations using different genome browsers. We can also do, as I said, de novo assemblies. In this way we can assemble the transcriptomes, and we can also study the expression profiles by counting those reads which are hitting to those specific regions. So in this way we are kind of quantification their gene expression. So here is RNA-seq. RNA-seq is a technique where we capture messenger RNA. We know that messenger RNA contains a poly A tail in the end. So the extraction methods, they identify messenger RNA using that poly A tail. This messenger RNA then gets converted into a DNA. We call it as a cDNA. And then those DNA molecules are fragmented, and then adapters are attached with it. So in this way we can come up with a library, we call it as EST, Express Sequence Tag Library, where adapters are ligated. So we submit that for sequencing, and sequencers, they give us those what we call them as sequence reads. So we take these reads and then we align them onto different references. So here we see a reference, and we have the whole genome in this blue part, and then we have the exons which are right here. So we can use the exons, since these reads are coming from the exons, so we will see that lot of reads and they will align on the exons. So those reads which are aligning on the exons, we call them as exonic reads. Whereas some reads that can go and just part of them, not all of them, but just part of them which are coming from the ends of these exons, so they have partial overlaps. So we call them as junction reads. So as a whole, we align those reads, and then after that, we count how many reads are aligned to these exons if we are doing this exome profiling, or the whole genes, which are how many reads are aligning to all exons present in that gene. So in this way, we count those reads together, and we can have an idea about the gene expression in terms of number of reads hitting to a gene. Genomics is the study of microRNA. MicroRNA are small molecules, about 22 nucleotides in size, and we have seen that so far in cancers, they have identified over 200 microRNAs. So microRNA study is really important if we want to study the gene expression. They are believed to be originated from the target genes. So if we sequence the microRNA from a cell, we can get those sequences, and we can then align them to the whole genome, and wherever they are aligning, we can suspect that their target genes are there. So for that, we might need to do some homology based studies where we can look into similarities of these microRNA into the target genes. So what we have seen in transcriptomics, that we can use these NGS techniques to study different kind of RNA, and RNA-seq is a technique where we can quantify the expressions of genes if we capture the messenger RNA complements, if we capture the total messenger RNA of the cells.